Yeah, whenever you're ready.
But not restart, right?
It didn't record it.
It didn't record it.
Yeah.
Okay. All right, so let's go over the concept questions from the central dogma. Number one, DNA replication is not directly involved in gene expression. Why is it part of the central dogma? Do you want this on there?
No, it doesn't matter.
So so I basically framed a lot of this in the context of gene expression because that's kind of what I'm interested in and what we're interested in as geneticists. But what is the central dogma? How is it different than just gene expression?
It also involves the storage of information.
Right, it's the storage of the genetic code and how that information flows from DNA to RNA to protein. Like how do we get that information from the blueprint for life, which is our DNA, all the way to the protein, right? And that involves DNA replication, right? All right, what is the difference between coding and non-coding regions of DNA?
The coding part of the DNA is the part that is transcribed, right?
Is coding, okay, so is non-coding DNA transcribed ever?
It's also transcribed, but it's not used later to produce proteins.
Right, so it can be transcribed, but it's not translated, right? So coding means that it's translated. So that means that you have these trinucleotide codons that code for amino acids, but you can have non-coding regions of the DNA that are still transcribed, right? Cool, and that could be like non-coding DNA, or we'll talk probably a lot more later in functional genomics about other regions of the genome that are not. Okay, so what are the three main steps of all processes described in the central dogma? So this DNA replication, transcription, and translation. What are the three main steps that all of them have? Yep.
Initiation, elongation, and termination.
Yep, initiation, elongation, and termination. Great. If you're interested in the amino acid sequence of a protein and can choose between obtaining the template or non-template coding sequence from the DNA, which would you choose and why? So if I just want to really quickly, like let's say I have like my little codon sheet and I'm like, okay, this trinucleotide codon makes this amino acid and I want to quickly figure out what are the amino acids in my protein, which strand of the DNA would you want to look at, template or non-template? So this is trying to get you to think about where the RNA is transcribed from, but what strand of DNA actually has the information you want.
Well, the RNA would match the non-template strand, right, with the substitution of the T and the uracil? Or is that opposite? I'm not confident in the answer.
So the template strand is the strand from which the RNA is transcribed, right? So the RNA is going to be the reverse complement of the template strand. And so it's not going to be like the sequence that you're actually interested in, right? It's going to, the reverse complement of the template strand is the non-template strand. So your RNA is going to be almost exactly the same as your non-template strand, except for, as you mentioned, the T's will be swapped out for U's. So yeah, you're on the right track. All right, cool.
So the non-template strand is the same as the sense strand?
Yes, so the non-template strand is called the sense strand, and it's also called the coding strand. And the template strand is called the anti-sense strand or the non-coding strand, because the RNA that comes off of it, that's like the complement to that, is the coding sequence. Yeah, cool. You were looking at coding mutations in a gene known to cause a certain disease. What type of mutation, synonymous or non-synonymous, would you think to be more likely to be the cause of the disease?
Probably non-synonymous.
Non-synonymous? Why is that?
Well, if it's synonymous, that would result in the same amino acid, right?
Right, so synonymous mutations result in the same amino acid. It doesn't mean that there's absolutely no chance that it has an effect because, as I said before, biology has exceptions everywhere, but it's more likely that a non-synonymous mutation, at least it's more interpretable. We can see that there's this amino acid change. Does anyone remember the two types of non-synonymous mutations I went over?
Of non-synonymous?
Missense and nonsense,
Yeah, missense, which is what?
change of amino acids,
Change in amino acid, and then nonsense, which is
early stuff.
early stop codon. Sweet. All right, what do we mean when we say the genetic code is degenerate?
Multiple codons coding the same amino acid.
Right, so multiple codons can code for the same amino acid. So we have 64 different possible combinations for codons, but only 20 amino acids. So that's what we mean. That means that the genetic code is degenerate. And this is not something I went over, but what might be a function of a non-coding gene? Can you guys imagine how that would be useful or used?
I don't know if this is the same thing we talked about, but we said at the end of the 12th period, you would add genetic code that's not used to like create any amino acids in the end, right? That would just be to protect the integrity of the rest of the strand. And
Yes,
Yeah.
but that's not considered a gene because it's not transcribed.
right.
But coincidentally, it uses an RNA template.
It does,
Okay.
yeah. So that RNA template would be a gene that's not
What about
transcribed.
the ones coding for like the T9?
Oh, that's a really good point. So we have like transfer RNAs and other types of RNAs that are not
Because they're
coding.
transcribed, but they're not really.
Right, they're transcribed in the nucleus and then they go into
Yeah.
the cytoplasm. Transfer RNAs bring the amino acids to the ribosome to make a polypeptide chain. That's a really good
The
one.
ribosome itself is a
Yeah.
lot of mostly RNA.
Ribosomal RNA, right? Any other function of non-coding transcripts that you can imagine? So remember, so DNA, if you have a single strand of DNA, it can be the complement to another strand of DNA, right? And so what we're learning a lot more now in human genetics is that you can have non-coding genes that bind to other RNA transcripts that can prevent it or enhance its expression in the cytoplasm. So these are sort of like this is just like a primer question for like the future modules and kind of thinking about what about these, like this, there's a huge number of non-coding RNAs and genes that we're like still learning the function of it. So I wanted to get your mind rolling on that. So, yep, that's it.
Yay, okay. Now we can be done.